Great. Thank you so much, Fruz. So my name is Rachel, and I'm a recent graduate of the Johns Hopkins School of Medicine and about to start as a staff scientist at the Hopkins Applied Physics Laboratory. It is my absolute pleasure to introduce Kathleen Morrill, who I met in Seattle last year as part of the Weintraub Award Symposium. And she's had an incredible research background. And so right now she is a senior computational scientist in genome analysis at Colossal Biosciences, who you may have heard of in the news as a company dedicated to preserving a record of genetic diversity of endangered animals and de-extinction of those we have lost, and bringing back the woolly mammoth as one of the headlines that you might have seen. She completed her PhD studying dog genomics in Dr. Eleanor Carlson's lab at UMass Chan. And she was interested in asking how pet dog genomic and paired behavioral data could actually be analyzed to get new insights into neurobiology and complex psychiatric conditions in humans. And so I'm really excited to hear what Kathleen has to talk to us about today, and I will just let you get to it.
Thanks, Rachel. Let me share screen. Okay. So, yeah. So back when we met each other at the Weintraub Symposium, this whole idea of large language models and these AI chatbots was still rather new, but we got on this tangent about how they were surprisingly handy for debugging code in a way that I didn't really expect. And at the time, I was finishing my PhD. I had learned kind of R on the job, and I was trying to make sense of my really messy code. And I realized that these were very helpful tools for kind of structuring code better, debugging problems, and that sort of thing. So we figured it might be neat if I shared how some of these models have been helpful for problem solving and crafting R code to tackle statistical questions in my own research. And I'm sure since that time that we met, many of us have become very familiar with these models. And since I'm definitely not into that whole, a lot of the hype around it, the prompt engineers, as people are calling them, instead of rehashing tips about how to use these models, I wanted to instead talk about why we find large language models and chatbots so helpful, specifically how models can be kind of analogous to what people call rubber duck debugging, which I'll talk about in a bit, and reasoning through language. I also wanted to talk about some handy but lesser known models for coding that aren't ChatGPT, including open source alternatives, how to access those either locally, remotely, online, and options for RStudio integration that we don't usually think about when we use these models. And I wanted to, in particular, explore a specific question, a scientific question from my own PhD research on dog genetics, which is actually where I'd like to start. So a little bit about me. I'm originally from Frederick, Maryland, so I'd still say that my home base, Our Lady, should be in Baltimore. Unfortunately, I'm in Massachusetts now, so it's a bit of a trek away. And my journey as a biologist and a geneticist started out from running experiments in the lab on things like bacteria, fruit flies, some plants. And I had some exposure to coding and statistics in college math courses, but it wasn't really until graduate school that I jumped into bioinformatics. And I started using dedicated programs for studying DNA and also trying to learn by doing with R and Python in order to run certain statistical analyses that just weren't suitable for some of the UI tools to run. And so I'm a perpetual learner, especially when it comes to writing code, and I always aspire to have better practices. My dissertation focused on a community science project about domestic dogs called Darwin's Ark. People would enroll their dog in our program, fill out surveys, and submit DNA samples for us to analyze. And that really was a dream dissertation. I'll talk about it in a minute, but right now I'm working on what's sort of my dream job as a computational genomics scientist at Colossal Biosciences, the so-called mammoth-making company. And as you can imagine, there's no shortage of interesting scientific questions to work on that lead up to that kind of moonshot goal, and while having the opportunity to help species conservation along the way. I am also into art and illustration. I love to draw. It's helpful with science communication, and my former thesis advisor was an artist too, so she inspired me to improve my kind of data visualization skills and get into more ggplot and other ways to visualize data. So doing a dissertation on domestic dogs was really fun. And I think that's because everybody wants to know more about our best friends who've been here by our side for so much of human history. And dogs are really this endless source of scientific questions, really beginning with the first one, which is what makes dogs dogs and no longer wild wolves. We can ask things like, what is domestication? Is it that the animals are friendlier? Is it the animals are less afraid of us? Is it that they take advantage of human environments like bed bugs? Are they domestic? Where dogs are kind of the quintessential domestic animal or domestic species. And we have a lot of questions about what factors led proto-dogs down this domestication path. How much direct involvement did we humans have? And what genetic changes solidified dogs as domestic and not merely just tame animals? And did that happen more than once? And those are all a bunch of deep dive questions. But what I was interested in for my PhD was the question of why do dogs do what they do? So there are all sorts of different lineages of dogs. What makes a dog a herding dog? What makes them good at that kind of job? What makes some dogs howl more than others? What makes beagles make this really weird vocal yodel? And other questions about, working dogs in general. And then there are also all these dog breeds that people have curated. So we have questions about what's their history? Where do they come from? How far back do their lineages go? What kind of diseases are common in these breeds? And what kind of genetic mutations are responsible that got fixed in their gene pool? And how can we resolve those? And then more general questions about how consistent are dogs in a breed? Does their behavior reflect their lineage of working dogs? Or are there some myths about breed personalities? Are all golden retrievers friendly and are all beagles super stubborn? And so canine genetics is a really great way to get at these scientific questions. And the first reason is that we can use genetics to trace the lineages of dogs to see how dogs today relate to one another, how many lineages exist. For individual dogs, we can trace their ancestry and their family tree. For groups of dogs like breeds, we can assess how much genetic diversity there is, whether any breeds are susceptible to diseases because they're more inbred. And once we've accounted for those factors, we can start to ask what kind of genes are associated with particular traits? What genes make dogs fluffy? What gene makes those dogs that look like mops have woolly fur? And are there any genes or regions of DNA that influence behavior, and to what degree do they influence behavior? And that's kind of the crux of my PhD research. And for any of those scientific questions to be answered, we needed a lot of data, which is why it's great that people love their dogs and they love to talk about their dogs. Because, of course, genetics aren't destiny, and so studying these individual dogs, including dogs that have these mixed breed ancestry backgrounds, was a great way to piece apart genetic factors from environmental factors and how they express their own individual traits. So then we come to the big kind of statistical question that I was left at the end of my PhD research, which wasn't so much which canine traits had a genetic basis. That alone is a kind of complicated question. In fact, I performed a lot of these studies on most of the traits that you see abstracted to the right. So I've performed genome-wide association studies connecting canine DNA to specific traits, like how sociable a dog is with people, how sociable they are with other dogs, how energetic they are, whether they like to play with toys, are they afraid of loud noises, do they beg for food, do they gain a lot of weight, are they allergic to anything, are they touchy-feely, and all these different traits that we can ask an owner about their dog. But each of those studies makes an assumption that those traits are independent of one another. And so despite many of these traits probably sharing genetic factors, in fact, we would say that something like fear-related disorders, such as a dog's noise sensitivity or thunderstorm fear, and separation anxiety probably aren't independent. They probably share some common genes that lead those dogs to be susceptible to developing a specific phobia, whereas a dog's experience and environment might manifest in different phobias depending on what they experience. And so with the data that we have, what kind of statistical approach should I use to find these kind of common traits, these overlapping traits that from a genetic level seem very similar, but might manifest differently. And so what I really wanted to be able to do was discern the hidden components that influence a dog's observed traits and what genes contribute to those components. So for example, in dogs, there is an age-related condition called canine cognitive dysfunction. It's also called doggy dementia. So this is a syndrome that includes observable symptoms like disorientation, lethargy, and incontinence. These observed behaviors share some common genetic roots connected to this latent syndrome that we call CCD. But it's also crucial to recognize that each of these behaviors might have independent genetic causes. For instance, incontinence could be caused by something like a kidney disorder or a bladder disorder. And that disorder itself might be pretty common in some breeds and might be connected to some genetic mutations. But that's independent of cognitive dysfunction or dementia. Moreover, other observable traits like a dog's body size can independently affect a dog's behavior. Larger dogs, like really large dogs, like Great Danes and St. Bernards, actually tend to be more lethargic than medium and small-sized dogs. And body size itself has its own genes that contribute to it. So if you're to perform an association study for the trait of lethargy, you might find the same genes you would find if you were to perform an association study of body size. Additionally, aging is not really a genetic trait, but it is a process, an environmental component that contributes to the development of canine cognitive dysfunction. And the aging process can be influenced by genetic traits like a dog's body size. So it all ends up being this really complicated web of influence, both environment and genetics, that are difficult to untangle. And when I look at the correlations between the association scores from the studies I performed as a thesis student, I can clearly see that there are traits that are not independent here. So then my question becomes, what are these clusters? Are they the same cluster that I would get through clustering the survey data alone? And what genetic markers do these traits have in common? So I ended up wrapping up my graduate school before getting to explore this question fully. But because I don't like sitting on data, I wanted to still try some things. But I didn't have any of my colleagues to turn to anymore because I was working a remote job. And initially, my inclination was towards dimensionality reduction, like a principal component analysis. But I was also seeing studies that were using all these other techniques that I was less familiar with, and I wasn't sure why and which assumptions were and weren't being made and whether it was really appropriate for my data set. So I ended up spiraling down this statistical rabbit hole and end up in like analysis paralysis, trying to decide what the right approach was, whether I was writing code according to the best practices, and I was maybe a little too lost to even begin asking friends or colleagues about what I wanted to do. So why not talk to a duck? So rubber duck programming or rubber duck debugging is kind of a name for a phenomenon where when you're really stuck on a problem, sometimes the best thing you can do is talk it out. Try to state your problem in clear terms, describe what your code is supposed to do, describe what your code is written as, describe what you're trying to do and what it's not doing. And sometimes that means going to a friend or colleague and talking to them and soon realizing while you're in the middle of explaining this problem exactly what you're supposed to do. And the other person didn't necessarily need to say a word. They could have just as easily, you could have been speaking to a wall, or you could be speaking to a toy duck. And so the term rubber duck debugging came about. And why I tend to think of these large language models or chatbots as rubber ducks is not so much that they are the duck in this situation, because they can be. They're certainly like a duck. They're an object that talks back. But the way that large language models operate is very much a thinking through speaking approach, where putting a problem into words and extrapolating on those words can converge on a solution. And that's kind of in its design. So large language models are these predictive models trained on a large collection of text tokens. That text token could be words, they can be parts of words like pre or post. And they can also just be code. Anything that can be put into plain text can be a token. And they have a very simple task asked of them. They're asked to predict missing tokens in the training data. So part of a training data could be something like the sentence, the quick brown fox jumps over the lazy dog. And in the training process, the model will have these tokens masked, and it's asked to predict what the most likely word is. So it's a very simple process. And then if a model is very small, it's not going to have a lot of patterns to recognize to fill these missing tokens in. But if the model is very large, it can become quite powerful. It can find patterns that would be really hard for you to pick up on that exist in its large corpus of training data. What was kind of new that happened about a year ago was not the auto-completion models, kind of like this duck here, but the instruction-tuned models that then took those models and fine-tuned them to be able to answer questions or complete a question with an answer, so to speak. Part of that large training corpus that's included in most of these models are things like Stack Overflow. So it's not surprising that they've actually picked up pretty well on how to solve coding issues or coding challenges. And what's also been observed about these models is that they can kind of seemingly reason through language. So the act of outputting language, they sometimes come upon really good solutions, especially when they're asked to do something like chain of thought reasoning, where they break down a problem into small pieces and then try to bring it all together. Do they actually reason? I really don't think so, but there's a lot of debate within the field as to whether you can get them to closely emulate human reasoning. And as far as accessing these models go, there is like the common ones that tech companies put out. They're very large. Running the models is not what's really expensive, although it can be very expensive. The real cost is in training these models, so that's why a lot of these very large models are operated under tech companies. And unfortunately, even the open source alternatives were usually trained by companies like Meta and just put out kind of out of goodwill. But a lot of those open source models are available to download, so you can run them yourself or through some sort of proxy that isn't, a subscription model that a lot of these are going to turn into. On the web, this website called Hugging Face collects a lot of open source models, and they have an alternative to ChatGPT, a web service called Hugging Chat that hosts some open instruction-tuned models like ChatGPT. So I'm going to show that in a second. It has a lot of good models, including one model that's fine-tuned towards coding tasks specifically, so it's kind of handy. If you have a really good computer and a compatible graphics card, like a NVIDIA graphics card, and a good deal of RAM, you could install and run most of these open source models, but they are all Python, so if you're not familiar with Python, you might get stuck. And I wouldn't say this is the most accessible way to run them. It has a lot of room for error, and I think your computer needs some like cooling system to handle it. So if you have a gaming PC, that might be appropriate. But there are options to run these remotely, so to connect to a virtual machine. And most of the open source models that are published also have a Google Colab notebook attached to them, where you can wait in queue for Google to give you a free virtual machine to run these programs on. It might just be delayed because other people are also waiting for these free machines. And then there is the kind of native support option where RStudio has partnered to some degree with GitHub Copilot, or at least they enabled GitHub Copilot, which is a subscription model through GitHub, an auto-completion model, not an instruction model, so that you can get predictions or suggestions on the fly as you're coding in RStudio. There are also other options for integrating LLMs with your RStudio environment, but most of them are just designed to minimize window switching. So they'll add a tab in the RStudio IDE where you can access a large language model, which isn't as useful necessarily as the auto-completion. So I'm going to share a different screen, my other thing here, and talk about a few of the models that I tend to use. So of course, there's ChatGPT, which is has its pros and cons and has its controversies, but it is very fast and it is free to use the 3.5 model if you're okay with them owning whatever data you put into them, which isn't always okay. If you work for a company, it's not the best idea to use these. But like I said, huggingface.co has these free alternatives, these open source alternatives in a UI that's very similar to how ChatGPT and other chatbots operate. And they have a few options here of publicly available models. Llama 2 is a very large model that was trained by Meta. This is instruction tuned, I think. And they actually made another version of it called Code Llama, which is that model, but fine-tuned towards more coding tasks. So this is the one that I tend to use. They'll give you some examples of commands to run, but you can just ask something like, what is the standard or Roxy gen documentation or something like that. And it's pretty capable. The other thing that I've been really interested in, but I don't know how much longer it'll actually be free to use, is Google's Notebook LM. And the nice thing about this system, it's not just the language model, but it allows you to make a notebook, allows you to add Google Docs, PDFs, and copied text, and kind of create an environment where you can have it cite specific sources that you give it in its answers, which is supposed to make it a little more accurate and a little more fine-tuned towards the task you're working on. So if I started a notebook here called Canine Genomics, I could tell it, this is me. I can give it some papers by importing some PDFs, and it will parse these text into a summary with some key topics, and also have the plain text for the LLM to be able to access. And what you would do is you would just select which sources you want it to use or not use, and your questions should be better tuned towards the things you're working on. So what I like about this for R development is if you are working with a newer R package where the documentation definitely hasn't made it into the training set for these models, but you still want it to be familiar with what you're doing and how it does it, you could just add the documentation here and use that as a specific source. So it's kind of nice because you build around it and it's more of a notebook type style of working with it. If you have GitHub and you happen to have GitHub Copilot, which is a paid subscription service, you can activate it in RStudio. There is an option under Tools, Global Options Copilot. And once this is activated, and I'll share an example of this as well, extra windows here. I currently have it activated, and it's an auto-completion model. It's not like a question model. I can't ask it like, how far away is the sun? It might know, but you pause and it'll make a suggestion. This is probably completely off. But what it's actually useful is something like load all files in directory starting with pizza _ and then files. You pause here and it speeds up your development to some degree. It's auto-completing and it has some context before and after. I found it's not that smart, but I don't need it to be. It's more about speed here. So each tool has a little bit of a different flair to it. So coming back to my problem, I thought that I'd just ask a bunch of large language models multiple times this prompt that I thought described my data and what I wanted to do in pretty simple terms. And all of these suggestions came up, all of them kind of along the lines of dimensional reduction or feature extraction, which wasn't surprising to me. I thought PCA would be the way to do this. And these two actually stood out because I've heard of these two approaches being used in human genomics before, didn't know a lot about them. In fact, I knew the first author of this paper from back before either of us started graduate school. So I knew that if I went down that rabbit hole and I got too stuck, I could always abandon the rubber duck and ask a real person. All these approaches seemed like they converged on this approach to factorizing an input matrix into some reduced dimensionality. All of them probably had specific assumptions about data distribution, independence, missingness that I could dig into more. And a lot of the times I wanted to re-implement these in R myself or use an R package when something like the SVD approach or MTAG might not have had scripts that were compatible with my data. So I actually ended up last summer trying to approach the SVD approach in my own GAR code and leaned heavily on chatbots to help me to do this. I think it didn't quite give me answers I would expect, and I can show that in a second. But this is a high-level overview of the kind of data I had and where I wanted to start from. So I'm going to go through a few examples of how I'd start exploring possible solutions to my question, how composing pseudocode might help, attempt some solutions and just try this on the fly. Let me share a different screen. So where's that showing? Oh yeah, what I was doing a little earlier to see if this would work out is I thought I could use this MTAG approach, and it turned out that it needed a data type I completely didn't have. And I actually ended up asking Notebook LM about it. I pasted in the PDF from this paper. I asked them from the theory section, can you tell me if this could be adapted towards my data? And they basically told me no, and it made sense because it was relying on this error matrix, and I had actually flattened this out by calculating z-scores. And I'm too lazy to go to the institutional server and get the really large data file that has the standard errors and the actual association effect sizes. So not going to approach that. It also wouldn't exactly give me what I wanted, which was which would be to collapse these three types of files into one, and rather than have these separate associations. So not quite the approach I was going for. But instead, I could ask them this question. Let me try a few different ones. Oh, exploration. So I asked, can it give me some sort of summary about the assumptions and appropriateness of these different statistical approaches? And I asked it to use train of thought reasoning. I don't know that it quite did necessarily. I think that these are very stochastic models, but sometimes they give you snippets of information that lead you down the right path. So I asked it to list out these assumptions and whether it's appropriate. And it basically said that SVD and PCA seem the most appropriate, and I'm inclined to agree with them there because these are both approaches that are useful for quantitative data. They're used a lot in these kind of matrices. So that's the approach I ended up going for. Another thing, I'm not going to try too many of these, but I'll just open up R here. I might also want to pause and ask if anyone has any questions or if they've been using chatbots in their own code development.
We do have a question in the chat from Sean, who says this is a very interesting approach on leveraging LLMs to aid academic logic thinking. Have you tried having the LLM write a quick prototype to experiment with the feasibility of a suggestion?
So I have not done a prototype so much as I have asked it before to give me a small tester data set based on a few examples of what the data should look like. And it's usually pretty good, especially if you ask it after it's made the function, like show me what I could throw through this function that might break it. And I've heard it has been pretty good about creating test cases for making sure functions meet all the criteria of being operational. So that's one good method to use it for, or one good approach. Let me see. So what I had here is actually loaded in my data, and I can show a little bit of what this data looks like. The matrix here is a dimension of 600,000 genetic markers by 185 traits. And I also have information about the traits and information about the genetic variants, including which genes these are close to. And if I run a PCA, I end up with something like, let's see. I end up with something that's actually a lot more sensible than when I did the structural variant, the single value decomposition approach. What I ended up with, if I summarize this by the PCA results by their highest contribution, you get things that look pretty sensible to group together. So at least one of these components, component 6, seems to have to do with how hungry a dog is or how prone they are probably to obesity. And if I look at the results of this, there are some genes that come up, but it would require a little bit more digging to see what those genes are. But I can, because this code is so messy, I can ask for some help with documentation. The most messy part of this code right now, I would say, is probably the singular value decomposition.
We have another question about, so when you're using ChatGPT or other chatbots, if you've had issues with hallucination and if you have any tricks for how to spot it, how to avoid it. And this person says that they've had it suggest functions that don't exist.
Yes, it commonly suggests functions that don't exist. I guess it's a lot of trial and error to see whether something's real or not. I have in the past asked it for packages. I think I asked it here. They recommended Psych, which I know exists. That's a popular one. I remember asking this one for some packages. So what's the best R package compatible with tidyverse that could be used for this? And you just have to verify each time. FAO MISC, I think it was misciting something here. Facto Extra isn't actually for running any of the multi-trait analyses. It's just for visualization and stats on it. Psych, I am familiar with, and a few of these too. Mixomics, I think it might have been an R package as well. Yeah, I think it's something to always keep in mind is that these things are very prone to making stuff up, and which is why I think having an environment where you're tethering it to reality, like this notebook LM, is pretty helpful. If you want it to be tethered to reality, it's best to provide it snippets of documentation on how to use something. It's helpful when a function doesn't have an example to feed the documentation and say, how would I do this? And another way I found it kind of helpful was for the TSVD approach that I was implementing. I didn't use Tanigawa's code as it was, but I went to the methods section and pasted the methods and asked them to parse it into kind of what the step-by-step would be. From the step-by-step, try to think of like what kind of pseudocode that would need, and then it makes it a lot easier to ask it piecemeal how to reconstruct what they did. And it's a little less hard with this because there's a little bit of, matrix math in here. But for more complicated methods papers that just give you an equation, and if you're a little less familiar with what that equation is doing precisely, it can be helpful to plug it in and ask a few times in a few different threads what it's doing to try to get a sense that is more in understandable terms so that you can recreate it in code. And I found that approach to be very helpful. And I don't know, it's really hard to think of an example on the spot. But I guess in the end, I ended up with some interesting candidates from just the PCA alone, which was what I thought was the more simple statistical method. I thought there would be some more complicated approach that I should be taking to find latent components. But if I actually take these, I think the variants from this ended up interesting too. Sorry, that's a little bit of a tangent. And I think the other thing is it's kind of a trial and error approach. It's not perfect. You always want to perform sanity checks. I think I still need to debug the SVD code I came up with from parsing out that paper because its components that resulted didn't make a lot of sense to me from someone who studied these traits before, how it clustered them, versus the PCA that made a lot more sense. And yeah.
Were you starting to go into using these to help like document your code and like best practices for documentation? Or
Yes. Yeah. So I didn't know about Roxygen before until I saw actually ChatGPT gave me out documentation in that format. I was like, what is that format? And it told me more about how to document code with Roxygen. So I think that that's particularly very handy. And it also showed me how to make 3D plots that I hadn't made before. So a lot of exploration that you don't necessarily find on your own.
were the 3D plots just using ggplot, or was that a package that you hadn't explored before?
That's a good question. Was it ggplot? Let me scroll down to it. I will share my screen again. So I had a 3D plot where I was kind of unsure how to explore these different values that Tanigawa had produced in his paper. So the contribution scores, the factor scores, and something else. Let me see if I can find it. Yeah, his paper had produced three different kinds of scores, these cosine squared scores, these contribution scores, and factor scores. And I just wanted a sense of what those were. And it's Plotly. I had heard of Plotly before, but I never made a 3D plot with it. So I described exactly what I was trying to plot. It's a very simple one. So I probably could have figured it out from documentation alone, but it allowed me to get kind of a quick sense for what kind of traits were popping out here. And they didn't really make sense to me, honestly. It's almost like a volcano plot here.
So if you were starting to use these for some of your like work at Colossal, where you have to be careful about, what you're inputting into some of these models that where your data might not be private, are you just foregoing using them at all? Do you ask them just for general questions? Like how are you, approaching them differently for like your data at Colossal versus your sort of, passion
Personal
project?
projects, yeah. When it comes to personal projects, like my thesis data has been published and is freely available, no holds bars. I'm happy to share whatever about it. When it comes to work data, I only ever ask it kind of questions about coding and how to do certain approaches and always mask what I am actually trying to talk about. So putting it into a toy example myself before I ever ask a chatbot to help me with code. I think that's the safest approach unless you're running these programs locally or if your company has some sort of institutional server where they run these models for you. I think that's the only safe approach. Because even from the way these models work, they pick up on patterns. And if they're retraining on data you're putting into it, even if you're trying to jump around what you're talking about, it's a pattern recognition model. So it's safer to not put in any industry data or even talk about exactly what you're doing, which is hard. And then I
Right.
haven't really been someone who's developed a full R package before, but I feel like if I were to start, it would be helpful to use chatbots to tell me exactly the best practices. So I've asked it before, how do people make these like nice automatic documentations for their packages? And it led me down some interesting paths that I didn't hear about before. I do wonder how many API keys ended up going into this training data. Oh, but as far as the conclusion of this goes, is it seemed like the association studies could be pretty well summarized, or a lot of the variants could be captured by 5 to 10 factors, at least in the structural value, the single value decomposition. In the PCA, if I bring up this is called the scree plot. I don't think it automatically made a scree plot, but 5 to 10 actually seemed rather low, but it also makes sense from the perspective that my data set itself wasn't that strong. It's only 3,000 individuals, and in genomics data, we need a lot more samples. So it's partly what I was so worried about was whether or not I had enough data to even do anything more interesting with it beyond the single studies. And I think I'll end there because I don't think it's all that interesting just for me to walk through playing with this data as much as to kind of get people's thoughts on like where do they see LLMs going? Do you think it's useful? Do we think that it's actually going to just weaken our development as our statisticians? So that's like kind of my back of my head is like I shouldn't lean on these too much, but they are handy.
Yeah, we do have a question about your thoughts on further expanding LLMs to help guide your hypothesis formation for a project. So, or providing methodology suggestions, giving feedback from the derived results, pointing you to other avenues. You'd kind of
Yeah.
shown some examples of these.
Yeah, the pointing me to other avenues I find it's what's most useful for is identifying all the gaps of knowledge that I didn't know were out there. And I think it's the embarrassment factor of, this may be something I'm less familiar with. I don't want to be completely stumped when I talk to someone about what that approach is that they're using. I want to get some base level expectations about what it is. You can ask questions without being judged to a chatbot, even if it's hallucinating to some degree, you can go and verify it. I haven't tried Chatter. I was excited initially. I would like, if I were to run Chatter, I'd probably try to install a local model and have it access that. I think that's one of its functions. You can either plug it into an OpenAI API and access ChatGPT, or you can actually connect it to a model that's running on your own computer. And then it really just adds a window where you can get prompts and get code out of it. But I haven't tried it out. I think it's nice, it's handy, but I think it's just as easy to have either be connected to a remote VM or have Hugging Chat open and talk in there. I haven't tried installing a local model yet, but I think that's the next step is to attempt that. The more parameters the model has, the more likely it's going to run very slowly on your computer.
Yeah, what kind of computer would you be running this on?
I'd say if you can run these models without a graphics card. But if you do, you need at least 16 gigabytes of RAM, is what I've heard quoted. I think the best alternative for someone that doesn't have a good graphics card at home would be to actually pay for an on-the-fly virtual machine from Google Cloud or Azure or something, because they're not as expensive as you think by the hour, as long as you're good about closing them out when you're not using them, and otherwise you'll rack up bills by the minute. But they're not actually that expensive to run. And it's possibly a cheaper solution than the upfront cost of building a really good computer.
All right, does anyone have any other questions for Kathleen? You can feel free to unmute yourself too if you'd like. Yeah, this was a really cool presentation. And of course, for anyone who came in a couple minutes in, this recording is going to be up on our YouTube. And we're going to have these slides up. And once Kathleen has a chance to sort of go through her code and clean it up a little bit, we'll be putting that up on our GitHub as well. Oh, we've got another
Yeah, that's a
question.
great question. Epistasis and pleiotropy. I'd say there's pleiotropy, such a wonky word. I wish I could have made this just about a science presentation, but it definitely needs more of a coding spin. But there's statistical pleiotropy, where it's the way that you're measuring the traits appears correlated, and therefore you end up with the same association. And then like true pleiotropy, I think is a lot harder to get at. True pleiotropy, an example of kind of statistical pleiotropy. We noticed that dogs shivering is highly genetic, or at least it had a very high heritability. And we're like, okay, so is there a gene that makes dogs shiver? Well, no, it's just that small dogs who have very poor thermoregulation shiver a lot. And the genes that make dogs small are so strongly associated with that phenotype that it statistically looks like it's causing them to shiver. But it's not that that gene makes you shiver, the gene makes you tiny, like a chihuahua. But pleiotropy is much more challenging to actually prove that something's happening. This would be the case of something like one gene controls two processes in the body. So maybe one gene raises your cholesterol, but it also results in waxy earwax or dry earwax or something like that. That would be something that's a truly pleiotropic gene. And it's hard to sort that out from statistical noise. Genomic studies maybe kind of like notoriously need millions and millions of samples to start finding associations. And you already know that a lot of those associations are correlated with other traits too. So it's an entire big statistical mess. I wish I had a really good example of true pleiotropy in dogs. Epistasis happens a lot. So the cause of corgis having short legs can be applied to any dog. So even if you had a Great Dane that mixed with a corgi, the short leg gene would kind of override it. They'd still be a large dog, but they'd have short legs no matter what, and that it's dominant. So one copy would mask any sort of tallness you could get from other genes. Definitely. Well, thank you, everyone. If you want to get in touch with me, my email is down below. I do have a Twitter account. Don't go on there very often anymore. Definitely a shout out to everyone that helped my thesis research, especially the participants and their humans. So two projects, if you have dogs to check out, would be Darwin's Ark or the Dog Aging Project. The Dog Aging Project needs a lot of support right now. They didn't get refunded this past grant cycle, and it's a longitudinal project of dog aging, so it's important that they keep funded. So participation in shouting them out would be really helpful.
Great, awesome. Thank you so much. This was a really great talk. And like I said, all of this talk and the materials are going to be available for you to check out. And please check out our other events that we have coming up this year. We've got a lot of cool things in the works. So we would love to see you all again in the future. So, and then reach out to us, of course, if you want to get involved in Our Ladies. So thank you, everybody, for your time. And thank you so much, Kathleen. This was
Oh,
really
thank you
cool.
guys. Yeah.